Hi, it's Mr. Anderson, and in this podcast I'm going to be talking to you about DNA fingerprinting. DNA fingerprinting was discovered by this man, Alec Jeffreys, at the University of Leicester, and is most widely used in forensic science as well as other uses of identification. Each of us has a unique genetic fingerprint or genetic profile that acts as a description of different parts of DNA that are unique to the individual. They are so variable that no two individuals in the world will have exactly the same genetic code in all regions. So the only exception would be in identical twins. A cell's DNA contains many non-coding sections which are known as introns, and a few of these introns make up the variable regions that are used for genetic fingerprinting. And humans have 10 of these. And within these variable regions there are repetitive sections of genetic code, and these sections are otherwise known as core sequences. And core sequences contain short tandem repeats of genetic code, and different people will have different numbers of code. So for example, you could look at this sequence of bases for a father and mother, and if I count this up, there are 1, 2, 3, 4, 5 multiple repeats in the father. However, in the mother, there are 1, 2, 3 repeats in the mother. So you can see that the length of the repeats is greater in the father than that of the mother, as well as the overall sequence. So if you were to cut the DNA at specific sections in the code outside of these repeats, so here and here, the length of the DNA fragment produced will vary because of the different lengths of repeated sections they are connected to. And since these regions can accumulate mutations without affecting the function, they will show considerable variation between individuals of the same species. The more differences in the bases, the more distantly related the individuals are. Sequencing is generally extremely expensive and slow, but fortunately there is a quicker way to compare non-coding DNA using the cheap and established technique of restriction enzymes and electrophoresis. And it so happens that non-coding DNA is mainly made up of long repetitive sequences. In fact, non-coding DNA accounts for between 95 to 99.9% of all of our DNA. And there are many different kinds of repetitive sequences. I won't go into them too much detail, but you can have satellite DNA, which is just any DNA region with a repetitive sequence. You can have tandem repeats, which is when a simple sequence of bases in a DNA molecule is repeated without a break. So for example, you could have 80, 80, 80, 80, 80, 80, so that would just be a 2-base repeat. You can have STRs, which are short tandem repeats, which are also microsatellites, which is a tandem repeat with a sequence of less than 10 bases. You can have a mini satellite, which is a tandem repeat with a sequence of 10 to 60 bases, or VNTR, which is a variable number tandem repeat, which is a region of DNA that contains a particular tandem repeat. And while everyone has the same repeating sequence in this region, different individuals have different numbers of repeats. So polymorphism, which is a range of different phenotypes of a characteristic of genotypes of a gene within a population, and in molecular biology it means a range of different DNA sequences at a particular locus. You can have restriction fragment length polymorphism, which is pronounced as RIFLIP or RFLP, and that's when restriction fragments of the same region of DNA have different lengths in different people. And just going into the process of DNA fingerprinting, it consists of 3 key stages, one being the extraction of the DNA, second being the polymerase chain reaction, and third, finally, being electrophoresis. So the first step of DNA fingerprinting is where samples of DNA are to be compared and amplified by the polymerase chain reaction. And these samples of DNA are extracted from the blood or sperm or anywhere where there is a complete DNA sequence. The PCR primers are designed to target and copy a region of the human DNA containing the VNTR region in this case, which is just the multiple repeat region. So as you can see, the VNTR region has been copied. In step 2, the amplified DNA of the VNTR region is digested by a restriction enzyme. the same recognition sequence should be present in the different samples, so that the number of fragments should be produced, the same number of fragments should be produced, but the fragments will be of different lengths, depending on the number of tandem repeats in each restriction fragment. So as you can see, this region has 1 tandem repeat and this has 4 tandem repeats, and you can clearly see that the restriction fragment in this region is smaller than that of this region. And finally, a fluorescent tag is added to all of the DNA fragments so that they can be viewed under UV light. And just going on to stage 3, where we use gel electrophoresis to separate out all these DNA fragments. And so the fragments are separated on the basis of their size using the process of gel electrophoresis. So DNA fragments are injected into wells and an electric current is applied along the gel. And it's important to note that DNA is negatively charged, so it'd be attracted to the positive end of the gel. So the shorter the DNA fragments, the quicker they move down the gel, so they're more lower down on the agarose gel. So DNA again, separated based on size. And generally a radioactive material would be added, which combines with the DNA fragment to produce a fluorescent image. So a photographic copy of the DNA bands is obtained, and therefore the pattern of fragment distribution can be analysed. Occasionally you may add a DNA ladder, which allows you to compare or work out the actual lengths of each fragment, therefore being able to compare the number of nucleotides that each fragment has. And if they're the same, then you can agree that 2 people are related if they're all of the same length. But by the same, I mean that they will have if they have the same length, they have the same number of nucleotides, therefore the same base sequence. I just wanted to finish off on the uses of DNA fingerprinting. So it can be used in forensic science, so to match DNA samples collected at a crime, so from sperm, blood, hair, or skin. It can be used to determine family relationships, so usually between a child and a suspect father, so that's just a paternity test. And since children inherit half their DNA from each parent, they'll inherit half their DNA fingerprint from each parent, so 50% of the bands should match their mother's band and 50% match their father's bands. And it can be used to prevent undesirable breeding during breeding programs in farms and zoos, as well as to identify plants or animals that carry a particular or desirable allele. It can also be used to determine relationships between ancient humans and modern humans, and to establish evolutionary relationships between different species. So that's it for my presentation, and please look at all my other videos. Thank you very much, and I'll just leave these photos on for